This is the behind the scenes.
The G protein linked receptor is the most common of all the transmembrane receptors and includes hundreds of different GPCRs. Although they are diverse, they all include one common structure, which is a single polypeptide chain that coils 7 times through the membrane of the cell. They are at times called the 7 transmembrane receptors for this reason. Each of these receptors have an extracellular and intracellular interaction site. The extracellular site is what allows the signaling molecule, or ligand, to bind to the receptor, and the intracellular site is where the G protein is able to interact with the GPCR.
At the beginning of the interaction, the GPCR has no signaling molecule attached to it. Because of this, the G protein is considered inactive due to how it is bound to one of the 2 guanine nucleotides it is associated with, in this case, GDP. When all 3 of these subunits are bound together, they form an inactive G protein. In order to start the interaction, a signal molecule must bind to the GPCR. When a signaling molecule binds to the GPCR, the G protein is activated and switches its GDP for GTP. The beta and gamma subunits now separate from the G protein, forming their own separate beta, gamma, and alpha complexes. The alpha subunit now acts as the G alpha and is considered active. Once active, it can diffuse along the membrane of the cell to activate certain enzymes that can then release a cellular response. When the G alpha has finished its task, which is indicated when the signaling molecule leaves the GPCR, it has to go back to its inactive state by binding back with the beta-gamma complex and taking in a GDP molecule.
When the G-alpha hydrolyzes, it goes back to its original inactive G protein state from the start, and this allows for the G protein to connect to another receptor for an almost infinite amount of times. Some proteins are actually able to prevent the protein from hydrolyzing. One of these proteins is cholera toxin and can lead to an uncontrollable amount of signaling. The effects of cholera toxin are more commonly known through how it leads to diarrhea because of excess amounts of fluids being created in the intestine. The uses of GPCR in the cell are used in signaling cells to their surroundings and whether or not they are in a habitable environment. Many of the senses, like sight, taste, and smell, are all moderated through GPCRs. One of the more well-known uses of GPCRs is how they regulate the production of the second messenger cAMP to the G protein's ability to control the enzyme adenylyl cyclase. The production of cAMP is important because of how it goes on to activate other intracellular proteins. One of these proteins is protein kinase A, which has the job of phosphorylating other target proteins around the cell. Although a lot of cAMP is produced, the signal does not last for a long time because it is quickly broken down through phosphodiesterase.
He's right here.
Oh, hey, I'll be home in an hour.